zinc oxide nanoparticles, carbon nanotubes, gold nanotubes, nanoparticles, silver nanoparticles, titanium dioxide nanoparticles, and chitosan. And nowadays we can see also PEG nanoparticles. Now here I would like to have your keen interest on the slide. Basically, it has been seen by Christina and the co-authors in 2012 that when we add silver nanoparticles to soil, there is an abundant effect on the relative relative abundance decrease. The decrease in the relative abundance of microbes. Basically, soil nitrifiers decreased with the effect of silver nanoparticles. You can see that ROS effect of basically silver Ag positive acts as an ROS, radioxydative species. It destroys the membrane of the microbe and the microbe dies. Therefore, you can see that application of these metal nanoparticles in soil science cannot be good for all the soil microbes because they can act as ROS for soil microbes and the microbial density will decrease. Therefore, now I will come to my doctoral research. From all these sides, you can see that nanoparticles have worked both beneficially as well as harmfully towards soil. Beneficially in terms of nutrient usage, in terms of increasing structural stability to soil, and in terms of decreasing the microbial growth. Therefore, uh, our aim was to synthesize fertilizers uh, that will be helpful in not degrading our soil microbial growth. Basically, metal nanoparticles destroy soil microbial growth. Therefore, we used an organic base. The organic base was for that we had first to synthesize and characterize the organic nanofertilizer, and then we had to see the effect of these on apple, basically red delicious. As all of you know that Kashmir is a valley of apples. Therefore, the first thing what we thought was to have to see the effect of these nanofertilizers on apple. The first part was synthesis of nanoparticles, and the chemicals used for the synthesis were chitosan, potassium persulfate, methyl acrylic acid, and formic acid. Chitosan is basically a polymer that is derived from sea shrimps. It is basically derived from chitin by deacetylation. Then potassium persulfate, it was used for polymerization process. Methyl acrylic acid is used to dissolve, and formic acid is used for maintaining the pH at which the nanoparticles will basically be produced. Now, what the process is? In this process, hello? Hello, ma'am?
Yeah, you are audible.
Okay, ma'am. Basically, what the process this PMMA, polymethyl acrylic acid, has acidic groups, COOH groups, and the chitosan has basically OH and NH2 groups. Which NH, no, you can see my pointer? Can you see my pointer? Hello, ma'am.
I yeah, we can see it. Yeah.
Okay. Basically, this amine side of the chitosan and this acid side of PMA coincided come in reaction with one another, and we have a soluble part of CSPMAA solution. Now, what basically happens? Preparation of chitosan nanoparticle using potassium persulfate as an initiator polymerization agent. Basically, K2S2O8. It acts as a polymerization agent with the help of methyl acrylic acid as a dissolution, and it has been already done by Hu and Zhang in 2002. The persulfate ionized attacks, basically when the chitosan and PMA react together, they form long polymerization chains. Then K2S2O8 basically cuts into those chains to form smaller chains and then wind them together to form the basically basic nanoparticles. The process like this, that dissolution of chitosan was done at 1200 rpm at a temperature of 72 degrees Celsius for 24 hours till the solution turned translucent. Like this, you can see here, translucent. Then addition of KPS, K2S2A8, what I also already told you that it cuts into those chains to form nanoparticles. To form the nanoparticles to produce a transparent solution was carried out at 700 rpm for 4 hours at a temperature of 70 degrees Celsius. Basically, it is quite important. You have to basically first develop this process. It will take you lot of time to develop the process, 2 to 4 months, because you have to continuously try to hit the right temperatures, right RPMs, and right pH for getting accurate size of nanoparticles what you have to actually get. Then loading of nanoparticles with nutrients. The first trial was with basically preparation of nanoparticles. Now I have to prepare nano fertilizers. For that, you have to load the nanoparticles with the fertilizers. Basically, when KPS was added, prior to that, we added nutrient. While nutrient loading was carried out, addition of nutrient was done before KPS addition, so that nutrient gets embedded into the nanoparticles. Basically, when KPS was added, it helped to produce the nanoparticles. But prior to that, we added nutrient into the solution so that nutrient gets embedded onto the chitosan chains, and the nutrient has embedded nutrients into it. Basically, the process was polymerization and micro encapsulation. Polymerization in forms of what Zhuang and other associates has done it like that, and micro encapsulation we added over it. We had a rectification to the process by helping micro encapsulation, means we added nutrient inside into the nano fertilizer. Then characterization process. Characterization was carried out by these instruments. It was basically FTIR, SEM, TEM, SEM EDS, and UV spectroscopy. Here you can see the FTIR graph. The variation in the peaks clearly shows variation in the bond resonation due to the addition of different groups. When we add different groups, different chemicals into the chitosan, firstly we added PMA, means polymethyl agarilic acid. The graph changed, means addition of polymethyl agarilic acid was there. Then addition of urea here, then addition of phosphorus, and then addition of KCl. Now, this is a scanning electron microscope which was used to basically see the size and shape of the nano fertilizers. This is basically sputtern. Uh, in in scanning electron microscope, you just need a conducting surface for getting the images. But as the nano fertilizers produced were of organic base, they were non-conducting. Chitosan in itself is non-conducting. So for that, we have to make it conducting surface. For that, we have to add a carbon film or a gold film on the upper surface of the bead. Here is the bead. We have the droplets of nanoparticle solutions, and to make this solution conducting, we have to add a carbon film with the help of these ion sputter. Carbon ion sputter. This was basically a carbon ion sputter. Basically, gold is very costlier, so we preferred to have carbon ion sputter. This is basically SEM images of the nano fertilizers. First one is of basic CSPMA, then second one with nitrogen, third one with phosphorus, and fourth one with potassium. This is transmittance electron microscope, and these are the images from transmitting electron microscope of nanoparticles. The basically single nanoparticle, nano fertilizer particle, single. Here you can see these. This is a basic grid at 200 nanometer. You can see how small these nanoparticles were. Usually the nanoparticles produced with nitrogen, free nanoparticles that will produce it without addition of nutrients like nitrogen, phosphorus, potassium. They were of the size 48 to 50 nanometers. While we had addition of nitrogen, the size was 78 nanometers. Then with phosphorus, it was quite closer to 98 nanometers. However, with potassium, the size was at 52 to 54 nanometers. This is the whole big grid. At 500 nanometers, you can see how much nanoparticles we have. There was quite a good load of nanoparticles into the solution. This is SEM EDS of chitosan and black nano. Basically, SEM EDS what gives you what? It gives you the amount of atoms or nutrients which are basically present in the solution. Here, we usually selected 10 pointers from the grade of SEM. Then we produce it as EDS, and the EDS gives you the exact number of count of the nutrients what we have inside the nano fertilizer. This is basically nutrient free nano fertilizer, nutrient free nanoparticle. Then when we had addition of nitrogen, here we can see the variation in the graph. Then this here is addition of nitrogen, and you can see nitrogen has been added into the, here you can see nitrogen has been added into the nano fertilizer. Then addition of phosphorus. Here you can see phosphorus has been added into the structure, and quite a good range of phosphorus in the nano fertilizer. Then when we added potassium chloride, KCl, because we usually use it, we use KCl as a fertilizer for all the crops for giving potassium, majority of crops. Therefore, Mr. Guzar raised hand. Oh, sorry. Basically, we added KCl. We add for addition to all the nutrients for supplying potassium as a nutrient to all the plants. We for majority of plants we use KCl. Therefore, you can see here potassium chloride. The chlorine has came into the count, and potassium has came into the count. Now, with UV vis spectroscopy of nanoparticles, you can see that it's a semi-parabolic graph. The semi-parabolic graph defines that your particles are nearly spheroid in nature. That means the nanopart fertilizers which we produce, they were basically of almost spheroid in nature. Then, uh, after production of these nanofertilizers, the important aspect of is that what was the fun of producing these nanoparticles? Why we have to produce these nanoparticles? The basic thing was that usually what we see in the soil system when we add nitrogen, phosphorus, or potassium, they are highly vulnerable to losses like volatilization losses, fixation losses, microbial losses. And usually with nitrogen, what we see, it is lost within 3 days in the soil system. Therefore, to find out whether our fertilizer will be able to produce, give something over a period of time, over a longer period of time in the soil system, for it we had to see the kinetic study. For kinetic study, the nanoparticles so produced were incubated at 27 degrees Celsius and were weekly examined for their particular available nutrient. For example, that one, the nutrient fertilizer that was loaded with nitrogen, we have to see what was the nitrogen coming out weekly. Then for phosphorus, we have to see the phosphorus coming out weekly, and the potassium nanofertilizer, we have to see what the potassium is coming out weekly basis. This is the graph of nitrogen, and you can clearly see the on first day, basically there were three nanofertilizers produced: nitrogen with 300 ppm of nitrogen, 200 ppm, and 100 ppm of nanofertilizer. The all the three same almost the same trend. However, the nutrient nitrogen nutrient fertilizer yielded more on first day. Likewise, on the second week, there was very less production. However, it continuously yielded to produce more nutrients with the time span. And at seventh week, it nearly exhausted. On seventh week of study, the nutrient fertilizer almost exhausted. Then with phosphorus, you can see the nutrient was available up to 9 weeks. And then with potassium, you can see it came to 9, 10, 10 weeks of kinetic study. Therefore, we can say that our fertilizer is produced, the nano fertilizer is produced, could provide plants with fertilizers for nearly 7 to 10 weeks, unlike those conventional fertilizers. And further effective to see on the apple plant, yeah, the basic treatments were like these: control, then T1, T2, T3, and the sprays were done at 2 stages. Basically, one was done before the onset of green tip stage, and second 60 days later, because we first assumed that our fertilizers will last for 60 days. Therefore, at 60th day, we gave second spray of nutrient fertilizers. And here you can see the effect of nano fertilizer over first nitrogen. And basically, nitrogen nano fertilizer, basically, first we were not knowing that up to which level of, up to how much concentration of nanofertilizer we have to produce. So to avoid phytotoxicity, we just went to the literature and produced the highest doses. For nitrogen, we produced only 300 ppm nitrogen. For phosphorus, we produced 50 ppm. For potassium, we produced only 200 ppm. But in the field study, it was shown that there is scope for producing more, because the highest treatment which we applied did show a significant effect over the prior treatments over 100. 300 showed quite good effect over 200 and 100. 50 ppm of phosphorus showed quite good effect over 40 and 30. Likewise, 200 ppm of potassium do show quite a good effect over 150 and 100 ppm of phosphorus. And these studies were carried out at 3 stages of apple: pink bud stage, pea size stage, and walnut stage. Therefore, the phosphorus percentage was likely more in phosphorus application than potassium was more with potassium application of nano fertilizer. Then we had a study over the sulfur, and it was quite pronounced with all of the three. Then calcium was quite good with potassium applications and nitrogen applications. And when it came to magnesium, the most interesting part in this research was in the field was that effect over magnesium, because potassium showed quite dramatic results. It was There was a negative correlation between these two things. That means when we added more of potassium, the magnesium in the plant is decreased. Unlike calcium, we added potassium, the calcium increased. However, when we added potassium nanofertilizers, the calcium in the plant is decreased. This is basically because of the negative correlation between the potassium and calcium and magnesium. However, magnesium is quite more predictable towards the negative correlations. Therefore, it decreases more obviously than calcium, as calcium concentrations in plants are quite higher, 10 times more than magnesium concentrations. Therefore, you can quite obviously see there that magnesium being quite small in the plant system, therefore effect of potassium nanofertilizers over the plants has been quite more. And with the boron, you can see the results are there. And yes, of course, with the physical properties, these quality properties, fruit drop, fruit retention, and fruit set, there were better results with the application of these nanofertilizers as compared to control. Firmness was induced, TSS was quite good, acidity was decreased, and with the color, in terms of L, A, and B, the color parameters quite had good effect with the help of these nanofertilizers as compared to control. Control was basically the fertilizer treatments that were given recommended by Stochastic K. Control was not absolute control, not only absolute control. It was basically control was made with the help of those nutrient fertilizers which were recommended by Stochastic K. Therefore, you can quite see the variation. That nanofertilizers quite have a good effect with the help of as compared to the fertilizers that were added conventionally, and yield was quite better with the help of these three nutrients. Uh, so this is the conclusion. And we successfully synthesized Cartesian nanofertilizers. Kinetic study of the nanoparticles revealed that application of N, P, and K nanofertilizers produced with the help to supply the nutrients to plant system for 7, 8, and 9 weeks. More precisely, nitrogen was able to yield for 7 weeks, 8 weeks with phosphorus, and 9 weeks with potassium. And application of N, P, and K fertilizers in conventional apple orchards showed significant higher results in all the doses of nutrient fertilizers for almost all the purposes. Therefore, giving a scope for having higher treatments. Therefore, that means we can have more than 300 ppm dosage in apple for nitrogen, more than 50 ppm for phosphorus, and more than 200 ppm for potassium. And the kinetic study was carried out in a priori to conditions that of plant system. That means at 27 degrees Celsius in BOD, the nanofertilizers were placed so that when we come into the plant, so that the conditions could be as good as like the plant system. However, these particles may reveal or react somewhat different in combination to the soil system. But when we will put these nanoparticles in soil system, they could behave something differently, because there are quite some different conditions. The pH will vary, the microbes will react to some act upon them quite differently, because chitin is basically a organic polymer. So it will be quite easily degraded by nutrients, these microbes. So the release of nutrients as good as in the kinetics were seen in kinetic study will not be as good as in the soil system, but they will be quite good, not as good as they are in the artificial those in the lab systems. Last but not the least, a positive ad was seen in the second year. The study was carried in 2 years, and it was seen that in the second year, the addition of the effects was more, because that might have been to the residual effect of the treatments that had been of the last year. So, thank you. I would like to thank you.
Thanks a lot. Thank you very much, Dr. Owais, for such a elaborate and well illustrated presentation.
Yes, ma'am.
Your presentation had valuable results and was enlightening to see the role of the nanoparticles. We owe you special thanks for being here, and we really appreciate that. And we will just check out if there are any questions or any query among the audience about it. You can just check the chat box for that. Oh, it is being said that thank you, sir. And some people may call you for more illustrations and elaborations regarding this presentation.
No
So
worries, ma'am.
I think participants are,
Everybody.
yeah. You can continue, sir.
Okay, ma'am. There is a question from Anisa. How is kinetic study done? It's quite simple. You have to basically first see the what you have to do. When I did the kinetic study, we just first incubated the things, the nano nutrient fertilizers in VOD over a period of time. The period which you have to see. For example, for 10 weeks, for 20 weeks, for 3 weeks, for 4 weeks, you have to incubate them. And at weekly interval, you have to get the sample out of the lab, then squeeze them with the help of pressure fertilizer filters. You cannot use a simple filter for getting the nutrients out, because nano fertilizers can easily get out through 48 watt paper, 48 size watt paper, 56 size watt paper, any other watt paper. They will easily get through there, because the pore size is quite bigger for nanoparticles. Therefore, you have to use 0.02 micrometer ranged plastic pressure filters for getting the nutrient out of the nano fertilizer. And then you have to quite do the analysis. For nitrogen, you have to go through nutrient analysis by colorimetry. Usually you have to go with colorimetry for all the nutrients. For nitrogen, you have to go with colorimetry. For phosphorus, you have to go with colorimetry. And for potassium, you can go with flame photometer. But the flame photometer should be highly precise. You can use equipment in RCRQA. They are quite precise in their expertise of determination. Therefore, you can use those for having kinetic study of the nutrients. I think there is only one question.
Okay, Dr. Owais.
Yes, sir.
It was very nice, wonderful, as already Dr. Sava has explained it. We loved it because our university has done such a wonderful job, and your contribution, of course, in this field. I think it was the first PhD thesis on nanoparticles being carried out in our SCOST. Of course, there were others to follow. Yes, maybe before that there was one MSc thesis, but first PhD research work, and wonderful research work. Now, I thank you, and of course, you are our student. I appreciate you, and hope you will shine more and prove your worth, because you carry a good worth. So we here conclude today's session, and we have one more lecture at 12. Let us, the same link will be used for that lecture as well. We can have a rest for 20 minutes, have a tea. Let us call it a tea break. Then at 12, we will be joining again for our another lecture. Goodbye.
Okay, thank you, sir.